So we kind of touched on this last time we talked about functional groups, that the functional group is what gives a molecule its properties. And we're going to focus on these 6 functional groups. These are the ones that we see most common and have the most influence on how molecules function in our cells. Okay, and take careful look. I mean, this one looks similar, and these carboxyl and carbonyl are the ones people often confuse because they are spelled so closely the same, 1-letter difference. We'll talk a lot more about carboxyl than we will about carbonyl, but all 6 do have a role in the functions of some of these molecules. So we'll start with the most common, I think, would be the hydroxyl group. Gets its name from the fact that it's just hydrogen and oxygen. But that combination of hydrogen and oxygen plays a big role in creating what we call polar or partially charged molecules. So here we have ethanol, which is the type of alcohol present in alcoholic beverages. Again, most molecules that have hydroxyl group, end up being considered alcohols. Not all, though. But this creates a polar nature to this molecule. And so far we've talked about that polar means it has a charge. But basically, you'll learn more about this in chemistry, but oxygen is an electron hog. It likes to steal electrons away. Yes, this is covalently bonded, and then the carbon and the oxygen are sharing electrons, but they're not sharing evenly. Oxygen's like the big brother that's not letting the little brother play with the toys often. Oxygen hogs electrons. So that makes this end of the molecule slightly negative. Not a full negative charge, but just slightly more negative than this side of the molecule, which is slightly more positive, which creates our polar nature of the molecule. As described over here, polar is a result of electrons spending more time near the oxygen than over on this side of the molecule. And because of that, we can form things like hydrogen bonds. You guys talked about water with Mr. Baumgars. Water molecules are also polar because they have an oxygen with 2 hydrogens bonded to it. The oxygen's slightly negative. And the hydrogens are slightly positive then, so we can form these weak hydrogen bonds to that side, and we can form hydrogen bonds to this side of the molecule. And that's why alcohols are easily dissolved in water. The water molecules can pull the alcohols apart, pull the separate alcohol molecules apart, and evenly distribute them throughout the solution. This also allows other organic molecules like sugars dissolve in water. Glucose has a bunch of these hydroxyl groups attached to its carbons. And so glucose and sucrose, all different kinds of sugars can easily dissolve in water because of the hydroxyl groups that make them polar. So that's a big functional role of hydroxyls that makes them polar, which makes them easily dissolvable in water, which we can make solutions. Very important for a cell. Okay, cells transport materials across their membrane via solution concentrations. So glucose and other molecules can easily move from high concentration to low concentration. They can diffuse because they are found in solution. They are dissolved in water. So that's the hydroxyl group. Let's get one in. Carboxyl, the next one. Sounds a lot like hydroxyl, has the same ending basically. We actually see a hydroxyl group in this functional group, but this whole thing is called a carboxyl group. Sometimes you'll see it written as COOH. It's kind of misleading because it looks like, well, the carbon's bonded to the oxygen, then the oxygen's bonded to the oxygen, the oxygen's bonded. It's not the bonding structure. The bonding structure looks more like this, where you have a central carbon here. What is this type of bond called? It's a double bond, so we're actually sharing 4 electrons instead of 2 here. And then you have this OH group right here. Oftentimes it's referred to as a carboxylic acid because this hydrogen can disassociate or separate and make whatever kind of solution that the molecules put in acidic. Lowers the pH.
So what organic means?
Organic means it contains carbon. Again, organic means contains carbon. So organic acids have carboxylic acid groups often attached to them. This is showing, I mean, we're not going to ever test you guys on this or anything like that, but this is showing how, this is acetic acid, it's just, 2 carbons in the molecule, but there's the carboxylic acid group. How that hydrogen right here can be pulled off makes this negatively charged, and the hydrogen's positively charged, so we do get a charged polar nature of the molecule from that, but we also get an acid when put in solution. Back when we studied proteins, what was the acid we were talking about that is the building block of all proteins? Amino acids, right. Amino acids have this carboxylic acid functional group attached to them, which is why they are called acids. So that's the carboxyl group. Not to be confused with the carbonyl group. What's missing from this carbonyl group? That's not the OH, no hydroxide part. So this does not necessarily form acids because there's no hydrogen to lose. There are 2 types of carbonyl groups. Ones that are in the middle of the molecule, like acetone. If you look, here's 1 carbon, 2 carbons, 3 carbons. This double bond oxygen, double bond O right here, is in the middle of the molecule. That makes a ketone, and those end in the -one ending. Or you have aldehydes that the carbonyl group's at the very end of the molecule. If you look, you can see the difference between this one and this one. Technically, it's the same number of carbons, same number of hydrogens, same number of oxygens. The arrangement's different, though. And that gives the molecule very different properties. Aldehydes will be polar. They will have a partial charge because that oxygen, again, is hogging electrons. But ketones, typically aren't because it's in the middle. So electrons are being pulled from this side and this side, leaving both sides positive. So it's not technically, actually not both sides positive, but the charges even out more in a ketone. We won't see them that often. You have all the functional groups to worry about. This is the one I'm going to stress the least. Doesn't play a big role in a lot of the molecules that we focus on in this class, so it's not as important of one. This one, however, is pretty important. It's the other part of our amino acid, an amine group. This one contains nitrogen and 2 hydrogens. What's interesting about amine groups, they act as bases because this nitrogen can accept a hydrogen. And when you take free hydrogen ions out of solution, you raise the pH, you increase the pH, make the solution more basic. So amino acids have a very interesting property to them. They can be both acids and act as bases. In fact, amino acids can act as buffers. They can stabilize the pH of a solution.
When you say bases, do you mean like the bases like phosphate sugar bases type of base, or just like?
No, when I say base, I mean more like as far as pH of a solution, it makes it more basic.
Oh, okay.
Like having a higher pH, like 11, 12. Wait, I know we didn't talk too much about acids and bases. I mean, I'm assuming you know a little bit from middle school what those are. Hopefully a little bit. You know, acids have a low pH, bases have a high pH. But the interesting thing about amino acids, though, is that because this can lose a hydrogen and this can gain a hydrogen, they can act as both an acid and a base, which makes it a good buffer. Another function that they play. But also, amino acids are building blocks for proteins. Basically, you lose hydrogen here and hydrogen in the OH there, and you can form a bond between the nitrogen and the carbon, and you can get a linkage bond, a peptide bond, to connect amino acids together. We'll talk more about that later, though. So that's an amine group. And then sulfhydryl. It's our only functional group that has sulfur in it, and it's just a sulfur and a hydrogen.
I don't think we have some.
I don't have that.
So like a phosphate.
It's not on that chart? Oh, can you add it?
Don't you just like?
Add a row?
Top, yeah. How do you?
Just like right click on a row and then, you or double tap if you're on the Chromebook, double tap with 2 fingers. And that right clicks. Or you hit Alt, I think. Alt click might be a right click too. Alt click is a right click as well. Insert row below. Or insert a, I mean, you can insert anywhere. There's no order to these really. Sulfhydryl, yeah. I can't believe that's not in there. Maybe I put the wrong one on. I know I don't teach sulfhydryl to the regular bio students, so maybe I gave the wrong one to you guys. But these are actually really important in the structure of proteins. We often see these in amino acids. We see these in the R groups. If you remember, when we talked about amino acids, we have this functional group here. This is known as the R group. And there are 20 different R groups. Amino acids like cysteine have this sulfhydryl functional group, and they act as cross-linking structures that help stabilize the protein. We kind of talked about it, but as a protein, we talked about proteins folding, right? Okay, this is what keeps the protein folded together. It's like the glue that holds the protein structure in place, preventing it from unfolding. And it's all these sulfhydryl groups that they form bonds with each other, and they kind of keep it all intact. I like to think of it like like a spider's web, like Spider-Man shooting his spider's web and like attaching things together. Kind of keeps it all folded up nice and neatly. So it forms these cross-linking, stabilizing bonds. I found this interesting, though, when I read it. The cross-linking of cysteines in hair proteins, so when guys might not have much experience with this, girls probably a little bit more, maintains the curliness or straightness of your hair. Straight hair can be permanently curled by shaping it around curlers. That's breaking and reforming those cross-linking bonds in your hair proteins. That's why you have to add heat, because the heat will break the bond and then allow the bond to reform differently. And so, any guys in here curl your hair? Do you, Fernando?
I know some people don't.
That's curly. Okay, that's how curling works. All right, phosphate. You have this one on there, right?
Yeah.
Okay, phosphate groups. This is the one that only contains phosphorus. We saw this in what major molecule? Where do we see phosphorus? We did see it in a lipid. I should say which two major molecules? Phospholipids has a phosphate group in it. What was the other one? What's this? DNA, right. Nucleic acids have a phosphate group, okay? Okay, it's basically phosphorus with 4 oxygens around it. One of those oxygens is double bonded. That's not important for you to remember, though. Usually found at the very end of a molecule, though. It's not going to be found in the middle. It's going to be found as attached to the end. Like this is glycerol phosphate. There's a long glycerol, and then there's a phosphate attached to it. Usually a molecule that has a phosphate group has the word phosphate in its name. Now, a big important functional part to this phosphate, well, there's 2 important functions. That one, it makes the molecule slightly negative, which means it can dissolve in water. It can be attracted to positive hydrogens in water and be easily dissolved. But a really important function is that it's involved with energy. We'll learn in this unit that if we break off phosphates, we can use phosphates as energy. This releasing energy part is really a big important functional role of phosphates. We touched on it when we talked about energy in the last unit. We talked a little bit about ATP, adenosine triphosphate. Those phosphates are involved in energy transfer. We need it for active transport. We need it to build molecules. We use phosphates as energy for a lot of processes that take place in our bodies.
You give us that on the next slide.
What?
Maybe
Yeah, I know.
Oh,
Isn't that exactly?
Yeah, it's exactly what we did.
Yeah, but you filled it out, so now you know it better. If you just copy this, you wouldn't know it. You'd be like, I can get a monkey to copy this.
Oh my.
Hey,
My
Yeah.
2-year-old, almost 2-year-old daughter can copy this.
She's smart.
I can copy.
Okay,
The last row.
but you can check yourself.
How do you spell releasing?
And sulfhydryl's up there.
Realizing energy.
These are the big biological importances of them. That's what I would focus on. I'm not going to, we're not going to have you draw them. Might have you recognize them. A lot of them are pretty easy. Sulfhydryl, it contains sulfur. Phosphate, it contains phosphorus. The tough ones, if you really look at the wording, carboxyl. Looks a lot like hydroxyl. Hydroxyl is hydrogen, oxygen. Carboxyl got a carbon into the mix. And like I said, the real tough ones are carboxyl versus carbonyl.
What is R? Oh, R is
R just represents, could be a long chain. So what I was trying to show here is that aldehyde, it's at the end, and the R kind of got cut off. The R should be right here. So it's a long chain of molecules that way, or of atoms that way, and this is at the very, it's like the caboose of a train. There are no cabooses anymore, but, okay. Whereas our ketone, there's molecules leading off this way, and there's part of the molecules leading off that way. This is stuck somewhere in the middle. Not exactly in the middle, but somewhere in between the two ends, which makes it less polar, not as charged.
But I thought amines could serve as lipid.
They can. It must not have been. It's just not written in that chart. I didn't put everything in the chart for you. Any questions on this? On these functional groups? Oh, maybe I'll give you like a functional group quiz or something next week or something like that.
Do it.
Not worth a lot of points, just to kind of just to see. If you,
Wants to change hydroxyl to gearbox.
it wants to. Google doesn't like carboxyl
I
as a
don't
word.
care, like, if it's not
Part
part
of
of
that,
it, then
it's
All right, so now we're going to move on to the actual molecules that we're interested in. We've talked about 3 of them already. We've talked about DNA. We've talked about proteins. And we've talked about lipids. Okay. What we didn't really talk about is the technical terms that we use to describe how these molecules are built. So when we talk about a building block, the technical term for that is a monomer. Mono meaning one. It's a single unit that can be connected to other units to build something bigger. Always make the analogy to monomers to Lego pieces. Each little Lego piece can be connected together to build something larger. So, for instance, in proteins, the monomers were amino acids. In nucleic acids, the monomers were nucleotides. Technically, there's no monomers for lipids because we have glycerol and we have fatty acid tails. Those are 2 building blocks that are not monomers. You don't link fatty acid tails in a row to get a lipid. So technically, lipids don't have a monomer. Polymer is just many monomers linked together. Poly means many. Polymers aren't necessarily macromolecules, especially like example are proteins. Proteins require several polymers to interact to make the bigger structure. So these are the macromolecules we're going to focus on in this unit. We're going to focus on carbohydrates. We've already talked about lipids and proteins and nucleic acids. We'll review those. The new vocab term will be carbohydrates, which are big, large polymers. So let's first talk about how we build and how we break down polymers. We touched on this in the last unit. We're going to touch on it again. We're actually going to test you on these in this unit. Are the 2 types of reactions that take place to either build a molecule or to break apart a molecule. So a condensation reaction is where you take the, for example, hydroxyl group from this side and the hydrogen from this side. We take these 2 monomers and we link them together. The result is, 2 building blocks that are now linked. Here's like 1, here's 2. But we also get water as a result, which is where we get the name condensation from. Condensation reaction gets its name from the production of water in the linking of those 2 molecules. So when we link 2 amino acids together, it's a condensation reaction. When we link 2 nucleotides together, it's a condensation reaction. When we link the phospholipids to the glycerol, that's a condensation reaction. Yes?
Do we have to know what type of bond it is when they link together?
Nah, not really. As long as you know it's a covalent bond, that's all I really care about.
I think it was in the I'm not sure. It might have been.
I know we do talk about peptide bonds when we talk about proteins. We're not going to talk about every different type of bond there is because that would just be way too much. It's not that big of a deal. Now the reverse reaction is called hydrolysis. Hydro referring to water. Think of the word lys as to split. It's splitting with water. It's the reverse of condensation. We take a water molecule and we add it to the polymer. We also have to add energy. It's not just going to happen with water. You can't put maltose and put it in water and then boom, you got glucose molecules floating around. You have to add some energy to it. But that's going to break the bond that's between, in this case, the carbons and the oxygens. Going to break that bond, and you will end up with, there's the water, there's the hydrogen from the water, and there's the hydroxide from the water, and then you get two separate molecules. These are two separate glucose molecules. So we essentially broke the molecule apart. So condensation connects, hydrolysis separates. So you should be able to, like on a test or a quiz, you should be able to recognize what reaction is what. I'm not going to say, okay, draw a condensation reaction. That'd be way too difficult. But you should be able to recognize, okay, I see two molecules are going in, we're getting water as a product, one molecule comes out, condensation. We connected those two molecules together, we got a polymer as a result, and we got water as a byproduct. Or we're breaking a molecule down. That's hydrolysis. All right, so we're going to move into our last major organic molecule we've not yet talked about, and those are carbohydrates. If you look at the actual word, carbohydrate, it tells you what's in it. We have carbo, which refers to carbon. And if you think of the word hydrate, that means water, which is H2O. So we have carbon, hydrogen, and oxygen.
Oh my God.
And in fact, it's even in that proportion. Generally, the ratio is for every one carbon in the molecule, there are twice as many hydrogens and the same number of oxygens. Roughly. Not exactly. So it could be written as this, where if we change the word N, or not the word N, we change the N to, like, let's say 6. If we had 6 carbons, how many hydrogens would we have? How many oxygens would we have? Good. This is glucose. If I change this to 3, how many carbons would we have? Oops. 3, how many hydrogens? How many oxygens? Okay. Again, not exactly all the time, roughly though. So what are the functions of carbohydrates? Generally, they're just our main source of energy. For example, glucose is the preferred molecule used by cells as its source of energy for cell respiration. Large carbohydrates like starches and glycogen are used as energy storage molecules. Your liver and your muscles pack away glycogen. It's just a bunch of glucose molecules linked together in a big polymer. And so when your muscles need energy, it just starts chopping the glucose molecules up to start burning for energy use. Now there are other carbohydrates that form structural molecules, and they're also used as cell identification markers. Like we talk about those glycoproteins and glycolipids found on the cell membrane. The prefix glyco means it has a sugar attached to it. Sugars are a simple name for carbohydrates. And so those act as identification markers for cells. So again, the main function we're going to focus on is energy. But there are other functions as structural and cell identification. All right, that is where I would like to stop for today. We'll pick up